Welcome. I'm Doug Sesserman, privileged to serve as the CEO of American Associates Ben-Gurion University of the Negev. And welcome to our live from Israel Ben-Gurion briefings, #webinar Wednesday. This is our 9th live from Israel Ben-Gurion briefing and our 14th since pivoting our communication strategy given the COVID pandemic. Today we are really pleased to bring Professor Simon Barak to you to talk about unlocking the secrets of plant life. And for those of you who are joining us for the first time, welcome. And for those who are joining us again, we really appreciate your participation in our webinars. As you know, this is about providing a glimpse into the great work at Ben-Gurion University and in Israel. Today's webinar is scheduled for 30 minutes. Simon will present for about 20 of those minutes, and we'll use the last 10 minutes to do Q&A. So if you're interested in asking questions, and some people have submitted them in advance, just click on the Q&A function. I'll be able to read your question, and we'll get them to Simon at the end of our webinar. So Simon, welcome. Just a little introduction of Simon Barak. So Simon made Aliyah to Israel in 1990. He's from London, England. You'll be able to hear that in his funny accent. He's a professor at the Jacob Blaustein Institutes for Desert Research at Ben-Gurion University for Agriculture and Biotechnology of Drylands at BGU's Jacob Blaustein Institutes for Desert Research, located at our breathtaking campus at Sde Boker, Israel, right in the heart of the Negev Desert. His research focuses on the understanding of the molecular mechanisms of plant tolerance to the stresses of harsh environments such as the desert, including stresses brought on by heat, drought, and high soil salinity. Professor Barak's team has discovered 2 genes that, when disabled, they lead to an increase in the plant's tolerance to multiple stresses. Professor Barak received his bachelor's degree in agricultural science from the University of Nottingham in the UK. He then made Aliyah to Israel and completed his Master's of Science in light control of plant gene expression, as well as his PhD in light and chloroplast signal control of plant gene expression at BGU's Jacob Blaustein Institute. Professor Barak did his postdoctoral studies in molecular genetics at the University of California in Los Angeles, UCLA. Tell us about your tremendous work, because it's actually quite remarkable. And Ronnie Strongen, who's our Chief Marketing Officer, she calls our tagline "From the Desert to the World," and I can't think of an area that describes that better than your science. Simon Barak.
Thank you very much. I'm just going to share my screen with you all. So just let me know that you can see this. Can everybody see that? Can you see that, Doug? Great. Okay, so first of all, good afternoon, everybody, or evening as it is here in Israel. And first of all, I'd like to thank Doug for his introduction. And also Doug and Melody and Liz and Cara and Ronnie and anybody else from ABG I might have forgotten for inviting me to talk to you about my research today. So you can see that what was shown to you was that the title of my talk was Unlocking the Secrets of Plant Life. And I actually added something onto the end of this, which is Unlocking the Secrets of Plant Life on the Edge. And the reason I did that is because my research deals with plants, or either with plants living really on the edge in extreme environments, such as this beautiful picture of a plant growing in the desert, or plants which might not necessarily grow in extreme environments, but are exposed to very harsh environmental stresses such as drought or heat or salinity. And I'd start by putting my research a little bit into perspective. So many of you will have seen this type of graph, and we're talking about here the problems of world food insecurity. So many of you have seen this type of graph with the increase in human population, and it's expected that the population will reach about 9 billion by 2050. And it's been estimated we need to increase our food production by between 50 and 70%. And that is absolutely huge. Now you might say to me, fine, well, just get more land under cultivation. But the problem is that most of the land that can be cultivated is being cultivated. And what's left are areas like this, arid areas that you can see in yellow here. And the problem with these arid areas is they're not conducive to growing our main crops because they're characterized by these harsh environmental stresses, such as if we look at the desert, drought and heat and high salinity in the soil and low nutrients in the soil. And so we have to find a way of engineering our plants to be able, our crop plants to be able to grow in desert areas. Now, even in areas that you wouldn't normally think of as having stresses, like say, for instance, parts of Europe or the United States, even there, 50% of all our crop losses are due to environmental stresses, be it drought and heat that you can see here, freezing, or salinity. Here you can see a field where they've over-irrigated with fertilizer, and you can see all the salts that are left after evaporation. This type of thing causes agricultural economy about $27 billion a year. So that's really, really a big problem as well. Now, the question is, can we actually make plants which are tolerant to stresses? And here you can see a stress-sensitive plant. This is actually a plant called Arabidopsis, and you're going to be hearing a lot about this plant in this talk, because this is like the guinea pig of the plant world, like the mice and the rats that you see in animal labs. And it's the first plant to have its genome sequenced, and many, many molecular tools have been produced from this plant. So you can see that it's very sensitive to salt, but you can see one of its salt-tolerant relatives, almost nothing happens when you add salt. So there are clearly mechanisms, maybe genes in these type of plants that we could maybe try and find and manipulate to make stress-tolerant plants. And that's really what my lab has been trying to do. So can we alter a plant's stress tolerance just with putting a new gene in, for instance, that we've found? And the answer is, in principle, yes. So here you can see normal plants, and here you can see the plants which have had a drought resistance gene put into them. And here you can see after 20 days of drought, they look pretty much the same. But once you rewater, the normal plant cannot recover, whereas the drought resistant plant does recover very, very nicely. So in principle, we can do that. But the problem is this is all very nice in the lab. But when you go out into the field, many times this doesn't work, because in the field, you're talking about multiple stresses simultaneously. So my lab has been trying to find genes which can give tolerance to multiple stresses. So how can you find a gene? How can you go about and try and find a gene which might be important to you? And one of the great things about working with Arabidopsis is they have collections which are publicly available, collections of mutant plants. And each of these plants has one gene inactivated. So you can imagine if I'm looking for a gene involved in salt tolerance, I might grow these plants with media with lots of salt. And then I look through the mutants and see if I can find something interesting. And here you can see a plant which looks, a mutant plant, which looks as if it's tolerant. So I've inactivated a gene here and the plant has become more tolerant. Or perhaps it's more sensitive, and that gene might also be important. The problem is that you have to go through thousands, if not hundreds of thousands of mutant plants, and maybe you'll find one or two. So this is really, really expensive and labor intensive. And when I first started the lab, we tried to find a method that we could use which would reduce the problem we have here. And this whole problem made me think of that book that we use with our children, Where's Wally or Where's Waldo, where you're trying to look in this whole crowd of people from just this one guy with a striped t-shirt and glasses. It sort of reminded me of looking for these one interesting mutant plant in these thousands of plants. And we thought to ourselves, well, what would happen if we were looking for Waldo, but instead of looking through all these people, we could actually reduce the number of people we look to only to those that are likely to be interacting with Waldo. Or if we go back to the plants, what if we could find genes which we know are likely to be involved in stress tolerance, then we would only have to order the mutants of those few plants, and we'd have to go through much less plants, and we would increase our gene discovery rate. So that's what we intended to do. And of course, I'm not going to disappoint you. There is Waldo sitting there. And by the way, if you really want to keep your kids occupied, you can color in Waldo, and then they'll be there for hours and hours busy. So how do we go about looking for genes which are most likely to be involved in stress tolerance? And to do that, we used a technology called microarrays. And basically what you have here is a chip. And this chip, all these little dots of DNA, it actually contains all 27,000 genes of Arabidopsis. It's like a little blueprint of life. And we can use these chips and we can ask the question, what are all the genes that are switched on and all the genes that are switched off when we give this plant, for instance, salt or drought or heat? And this is what we did. And using some sophisticated computational biology, which I can't really have time to show you, we managed to narrow down the genes which are most likely to be involved in stress tolerance. We ordered the mutants only of those genes, we tested them, and indeed we managed to find mutants and genes which are important in stress tolerance. So for instance, here you see Arabidopsis, the normal plant, grown on some gel which has high levels of salinity. And what you can see here is that this is causing the plants to die. You can see that the leaves are becoming white here. Whereas the mutant, which we called STRS1 or STRS2, is really tolerant. You can see the same with the high temperature. Here are the normal plants, and you can see that the mutant plants are more tolerant to the heat. So we did a lot of work trying to find out what was going on here. And what we found out is that in the normal plants, when you give them stress, the stress tolerance genes are activated. But when you give the mutant plants, which have had the STRS genes, the stress genes, inactivated, you give them stress, those stress genes are switched on even more. They're massively switched on. So we're wondering, what is the function of these stress genes, these STRS genes? What's going on here? Now, very often when we want to understand the function of a gene or the protein that the gene produces, because most genes produce proteins, we ask where within the cell is the protein localized. So for instance, if we find the STRS protein in the nucleus, we might have a hypothesis that we can test that it's involved in controlling gene activation. Or if we find it's localized in the mitochondria, which are the energy factories of the cell, we might hypothesize that it's involved in energy production. So how do we know where, how can we find out where the STRSs are in the cell? And to do that, we use this really neat technology called green fluorescent protein, or GFP. This is a gene from jellyfish. And if you put this gene, for instance, into a fruit fly, the gene will make the GFP protein. And when you put the fly under UV light, it will glow, it will become fluorescently green, and it doesn't harm the fly at all. So how can we use this green fluorescent protein to know where our STRSs are in the cell? So what we did is we took the STRS gene, we fused it to the green fluorescent protein gene, and we introduced that into the plant. So now we'll get a protein, STRS fused to the GFP protein. Wherever the STRS goes into the plant, into the cell, it will drag the green fluorescent protein with it. There you go. And wherever it is within the cell, that cell will now become fluorescent. That part of the cell will become fluorescent. And that's exactly what you can see.
We're already getting some really good questions. So
go ahead.
If you do have questions, just click on the Q&A function. The one question, Simon, is how do you actually get the gene into the plant?
So to get genes into plants, what we do is we use a bacteria called Agrobacterium, which in nature causes tumors on plants. And the bacteria has a little piece of circular DNA in it, which has these genes, these bad genes, which cause tumor in plants. What we do, and so how does it cause the tumor in the plant? It actually takes those bad genes and inserts them into the DNA of the plant. So what we do is we remove those bad genes, we put whichever gene we want, and we allow the bacteria to do the work for us. It actually will insert our gene into the DNA of the plant. We usually do it into cells, and then we can regenerate whole plants from individual cells. So that's a basic idea of how we get genes into plants. Any other questions, Doug?
Nope, keep going.
Yeah, okay, so I hope that answered the question. So here you can see the two cells where with this green fluorescent protein in STRS1. So you can see that this is the nucleus of the cell. And if we zoom in on the nucleus, you see it's all lit up, and there's this big blob also lit up, and this is a compartment even within the nucleus called the nucleolus. But when we examined even further into the nucleus, we found that the STRSs are sitting in regions of DNA, which are lit up here, which are silenced. This is called gene silencing. So what is gene silencing? So we all have different cell types in our body. I hope most of us have brain cells, and we have liver cells, skin cells. One of the differences between these cells is that in some cell types, certain genes are completely switched off, they are silenced, and in other cells, other genes are silenced. And we were able to show that these STRSs are absolutely involved in gene silencing. So what we found is basically when you give stress to our Arabidopsis plant, it will activate its stress tolerance genes. But our STRS1 and STRS2 genes, they're there, they say, hang on, maybe this isn't a real stress, maybe it's a temporary stress, and they act like a brake. Don't activate all your stress tolerance genes, or only activate them slightly, costs a lot of energy. But when we mutated those genes in those mutants when they're not there, we are effectively removing that brake. And so when we give the environmental stress, we remove those STRSs, we remove the brake, and the plant massively activates its stress tolerance genes. And so this gave us an idea for a great hypothesis. We thought to ourselves, gene silencing, maybe if we mess around with gene silencing, maybe if we loosen gene silencing a little bit, this could be the key to creating, or a key to creating stress tolerant plants. And so this is what we did. And so here you can see Arabidopsis grown in high heat, 45 degrees. You can see that most of the seeds didn't even germinate. And if they did, the plants look really poorly. And now I'm going to show you some plants where we slightly loosened gene silencing, and you can see that they are massively tolerant to heat. So this is something that we are trying now to work very, very hard on, looking at gene silencing and stress tolerance. Now, I've mostly been talking about this plant, Arabidopsis. And the problem with Arabidopsis is that it is actually a stress-sensitive plant. And if we want to find new genes and new mechanisms, we should really start looking at plants which are naturally tolerant to stresses, such as plants living in some of the most extreme environments in the world, whether it's deserts or in the Antarctic or in alpine areas or under the sea. And maybe they will have unique mechanisms and genes. Now, many of these plants are quite difficult to work with. So what my lab did and several of my collaborators around the world is we said, okay, why don't we look for relatives of Arabidopsis that are extremophyte plants living in extreme environments? And so we began working with these species. One of them is Eutrema, which you're going to see quite a bit of now. This is in China and in the Yukon in Canada in very, very salty soils. So it's very tolerant to salt. And we also work with a plant called Anastatica, the true rose of Jericho from the Negev desert. And we can use now all the molecular tools that have been developed for Arabidopsis to look at these relatives and see why they are tolerant to stresses and Arabidopsis isn't. So I already showed you this slide of Eutrema. Here's Arabidopsis, very, very sensitive to salt. Here's the Eutrema, nothing happens to it. So what unique mechanisms, genes can we find in Eutrema that allows it to do this? If we look at Anastatica from the Negev desert, it's really tolerant to heat. So here you can see Arabidopsis and Anastatica grown at 45 degrees. And you can see that after 2 hours, Arabidopsis is dead, but Anastatica just keeps on going. It's also tolerant to low nutrients in the soil, but I'm not going to go into that right now. So with the time I've got left, I just want to show you just a few things that we've managed to find that make these plants, these extremophyte plants, living in extreme environments special. So for instance, here you can see Arabidopsis and Eutrema. And we have added salt to the soil. So these little blue dots are sort of salt molecules, actually sodium from sodium chloride. Now, both of these plants have pumps, protein pumps that can pump out the salt and prevent it getting into the plant. With Arabidopsis, that pump is not very efficient. So what you see is many of the sodium ions, they actually accumulate in the plant. Only some of them get pumped out, and this damages the plant. But with Eutrema, it has a pump which is very, very efficient and pumps out all the salt. So if we're now thinking about making salt-tolerant crops, we should start thinking as well, for instance, of making sure they can pump out the salt. So why is the Eutrema pump so much more efficient? And we thought to ourselves, well, maybe something going on at the level of the genes, not just with the pumps, but maybe with all different or many different salt tolerance genes. So we said, okay, let's have a look at what's going on with all the genes. Which are all the genes which are switched on and switched off in the salt-tolerant Eutrema versus Arabidopsis? And we used some really high-tech technology called RNA sequencing. And we found some really, really interesting stuff. So this is Arabidopsis, salt versus no salt. And in this graph here, everything above this red line, these red dots, everything that's a red dot above this red line, are genes that had significantly increased activity when we added salt. And all these genes below the red line are genes that had significantly decreased activity when we added salt. So you can see about 3,000 genes in Arabidopsis increased their activity when we added salt, and 4,300 around decreased their activity. But when we looked at the
Simon,
salt, yes,
we're getting a really good question from one of our anonymous
go ahead.
attendees, because you're talking about genetically modifying plant structure, right?
I am. Genetically modified organism, yeah.
Okay, so we'll talk about the ethics on that a little bit at the end, but
yes,
this question is in particular, can these methods that you're describing be applied to food crops specifically? And if so, which crops?
So yes, these methods, actually these, for instance, this sequencing method, one of the great things about it is it can be actually applied to any plant and crops as well. As far as putting genes into plants, transforming the plants with new genes, that depends on the plant. Some plants are very amenable to that, and some plants are much less amenable to that, and we have to find different methods in order to put the genes in those plants. So I hope that answers the question for now, and I'll be happy to expand on that if anybody wants to in a few minutes. So is that okay, Doug? Anything else?
No, no, I think it's great. Wrap up. We're getting some other really interesting questions.
Cool. So we're almost finished. So just to go back, so in the Arabidopsis, we get lots of genes, thousands of genes with increased activity or decreased activity when we give salt. Now let's have a look at the salt-tolerant plant, this Extremophyte plant. It hardly responds. We only get 118 genes with increased activity and only 435 genes with decreased activity. So these Eutrema genes are hardly responsive to the salt. And what we found out is that in Arabidopsis, the same genes that are activated by salt in Arabidopsis are already activated. Even without stress, they're already activated in the stress-tolerant plant. And the genes which are deactivated by salt are always deactivated already in the Eutrema plant. So it's primed for the stress. And now when we're thinking about making stress-tolerant crops, we can think not just about genes, putting unique genes or different genes in them, but actually changing the way those genes are activating. Taking the pieces of DNA, the molecular switches in the DNA, and using those to change the way genes are expressed or genes are activated in crop plants. Okay, the last thing I'm going to show you is this. And the title of this plant was Unlocking the Secrets of Plant Life. And I think when you're trying to unlock the secrets of any life, one of the great things to do is to have a look at the genomes. All the genes and all the DNA in a plant, because this gives you into a window really into the blueprint of life. So I'm going to just show you one example of looking into the genomes and comparing the genomes of Arabidopsis, the salt-sensitive plant, and its salt-tolerant relative. So both plants have a pump called HKT1, a gene which encodes a pump called HKT1. This actually pumps salt or sodium into the plant because the plant does need some sodium to function properly. And when you look at the genome, you can see that Arabidopsis has one copy of this gene. But when you look at the salt-sensitive relative, the salt-tolerant relative Eutrema, in the process of evolution, it's actually got the same gene, but it's actually duplicated that gene twice. It's triplicated the gene. And when you have a look at what happens with this gene when you give salt, the original gene in Arabidopsis, you give salt and it's deactivated because you don't want salt being pumped into the plant because that will damage it. Too much salt. When you look at the Eutrema, you look at the exact same copy, the same thing happens. It's deactivated. But when you have a look at this copy, it's actually activated by salt. And when you delve right down into the sequence of this gene, you actually see there's a little mutation there. And this mutation has turned this pump from a sodium pump into a potassium pump. And plants which are under salt stress really need potassium. So that's just one tiny thing you can find by looking at genomes. They really are a window into the genetic basis for adaptation to extreme environments. So just to tell you that we don't just use plants that live on land, we also use plants under the sea. These are seagrasses. They're very salt tolerant because they're under the sea, of course. We've got this fantastic setup with my collaborator in the Dead Sea and Arava Science Center, Dr. Gideon Winters, of aquaria where we can grow these plants. And we've just started a big project called Seagrasses, where we're trying to look for salt tolerance genes from seagrasses that hopefully in the future we can make salt tolerant crops from. And of course, I have to, before I stop, is to thank the superheroes of my lab, which are the technicians and students and postdocs who I've had over the years. I've really been lucky. They've been absolutely amazing, and they're the people who really do the work. So I will leave you with this wonderful picture of the Midrashet Ben-Gurion, and the Jacob Blaustein Institutes for Desert Research. I hope that any of you who haven't been here will come and visit us and see some of the amazing research that's going on. Come to visit David Ben-Gurion's grave and of course see the magnificent scenery of the wilderness of Zin. So thank you for listening, and I'll be happy to take questions.
So thank you very much, Simon. That was terrific. We're almost out of time, but I'm going to take the moderator prerogative and we'll extend for a few more minutes because we really did get great questions. And
Sure.
For those who have asked questions, if we don't get your questions answered, we'll do our best to get back to you afterward. I'm going to try to combine a couple of these for you, Simon. So David Breslauer, one of our board members, Michael Ozer, who's also very involved with us from San Antonio, they're both asking about commercial applications of this research. David specifically is asking about if a plant transfers salt back into the soil, what's the implication for commercial plant growth in terms of sparser density, et cetera?
Okay, so we'll start with the first question about the commercial applications, right? So, for instance, when we did our computational screen trying to find genes, we actually did this in collaboration with Bayer Crop Science. So Bayer Crop Science are one of the three big companies that deal with genetically modified crops. And they were very interested because they felt that these were new genes that had not been found before that could increase the tolerance of plants to harsh conditions. And so what we want to do is especially now with the new gene editing technology, which is not considered a GMO, we can try and manipulate the genes in the plants to make them stress tolerant, grow them in arid areas, and of course that has huge commercial potential, and grow them in regular areas where we get these huge losses due to environmental stresses. So there's definite commercial application. In fact, the Seagrasses project is a joint project with the Ministry of Agriculture of Israel and a venture capital fund who also see commercial promise in Seagrasses. For the other question about pumps back into the soil, the salt back into the soil. Well, you're talking about minute amounts of salt compared to all the salt in the saline soil. So it's absolutely negligible the amount of sodium ions that the plant is putting back into the soil, but it's not negligible for the plant itself and the damage that those sodium ions are doing in the cell.
Thank you. So we're getting a really good question from Hadassah Weiner, and Hadassah, thank you for joining us. She's asking about the relationship between salinity and food quality. So I know in particular, cherry tomatoes are a great crop in the Negev desert. Can you just speak in terms of the relationship between salt
Sure.
in the soil and taste of the food?
Sure. So when you give things like salt or many other chemicals or many other stresses, it causes the plants to produce many sorts of different compounds to protect the plant. And some of those protective compounds will change the way that the plant tastes, the way the plant looks and feels, the texture of the plant as well. And so that's why when you give salt, for instance, to cherry tomatoes, it causes the production of defensive compounds, and those are or sugars, which also help in defense, and those are why they become very, very sweet.
Thank you very much. I think a tomato even tastes better than a tomato.
Tomato, potato, let's call the whole thing off.
So let's wrap up with one closing comment from you, Simon, on the topic of GMOs, and we've gotten a couple of questions. So we're talking about genetically modified organisms. It's a controversial subject. Mm-hmm. This is at the heart of your research. You're a nice Jewish boy who went to Hebrew school, then you became a scientist. Just talk a little bit from your perspective around the ethics of GMO.
So what I'd like to do, I often get this question, so I actually made sure that I had just one slide just to show something to everybody that always tends to shock people. They don't really think about it like this. So if I may share my screen once more, okay? And the reason I want to show you this is because, here we go. So in fact, if you think about it, all our food is genetically engineered. And I want to show you this picture here. This is corn 7,000 years ago. It's tiny, about the size of, I don't know, half a pencil, okay? And this is corn 500 years ago. This is before breeding. This is before genetic engineering, okay? This is just domestication, where farmers would take a seed of a bigger cob that they found in the population, they'd resow it, and gradually the population was fixed and the cob became bigger and bigger. If that isn't genetic engineering, I don't know what is genetic engineering. Even if you think about breeding, that is genetic engineering. It's just we're taking all the genes of one plant and all the genes of another plant and we're mashing them all together rather than taking a single gene. So that's one thing. The second thing is that there's been a lot of disinformation out there about genetic engineering, and partly the companies are to blame for this, and partly the environmental groups as well. Now, I don't blame the environmental groups. They've actually done some good work making sure there's lots and lots of checks on these genetically modified organisms before they go out to the market. But one of the problems is, I'll just stop sharing my screen. One of the problems is that when you have a gene, you find a gene, to get it to market takes about 13 years because there have now so many of these tests, some of them completely unnecessary, that only the large companies have the money to do it. So all the small companies have been eaten up. And now you only have three companies, really, three major companies. You have Bayer Crop Science that just ate Monsanto, you have Syngenta, and you have DuPont Pioneer. And to me, it's much more worrying to have only three companies controlling our food supply for profit than it is for having the actual technology. The technology itself has been proven to be safe. Mark Linus, who was the one from the Friends of Earth, who was a major campaigner against genetically modified organisms, has agreed with this. And in any case, now with the new gene editing technology, these are not considered GMOs because we're not putting foreign genes into them. So I will just end my answer to that question with a quote, which I always like to give, because there's also a cultural element to this. And the quote is as follows: in America, products are considered safe until they're proven risky. In England, products are considered risky until they're proven safe. And in Sweden, products are considered risky, especially if they're proven safe. So I think some of the times this is also a bit of a cultural thing. Europe ban it, America allow it. So that's my take on GMOs.
Well, thank you. I think those are great words to close this webinar. I want to congratulate you, Simon, on holding the audience. Even though we went a little bit long, you may be the first scientists to present on our hashtag Webinar Wednesday live from Israel Ben-Gurion briefing, where the participant numbers stayed and actually grew throughout the whole webinar. So,
Thank you.
Yasher Koach to you.
Thank you
Those
very
who are
much.
interested in more and just can't get enough, in 2 weeks, we'll have another hashtag Webinar Wednesday live from Israel Ben-Gurion briefing on the topic of cutting edge cancer research, the amazing work going on between Ben-Gurion University and Soroka Hospital. So please join us on September 2nd at noon Eastern for that. David Ben-Gurion, the founder and first prime minister of Israel, and who I call the #2 Jew all time, and also the guy whose name is on the door of our university, like Simon himself, was an ardent Zionist, and he once said that Jews will reclaim their ancient land in Israel, not by argument or by invoking historical precedent, but by their labor, by creating an enduring, fruitful home for themselves where previously there was nothing. I don't think anything describes that work better, really, than yours, Simon, and the incredible work at the Jacob Blaustein's Institute, literally bringing food from the desert to the world. Thank you, everyone, for joining us. Be safe and stay healthy.